So very nice to be presenting here about depression. That's the disorder that I study most in my last 25 years. A study with a huge burden. It is in the top 10 of diseases that the World Health Organization lists as most impactful diseases on our population. And I was involved in a Lancet commission on depression. We published a paper on that in Lancet a few years ago. And I think the bottom picture that we showed in that paper is really very depressing figure, because it really shows that despite the fact that over the last 3 decades, there has been lots of changes to treatment of depression, to improving and increasing healthcare for people with depression, we are still not able to change the impact that depression has on years lived with disability and disability-adjusted life years. So the burden of disease has not been decreased. And I really wonder why, and I think it's the question that we need to address as a community research as well as clinicians. And I think one of the major features that potentially could help explain why we are not able to do a better job is the fact that depression is a very heterogeneous condition. And this heterogeneity is hindering research because it may lead to inconsistent findings. And it is also hindering treatments, because that may be a reason why we have small effect sizes of the standard treatments, and that certain treatments clearly don't work for all individuals. So one size doesn't fit all. I think it's important for us to recognize the heterogeneity, but also use it to examine and better understand what is going on in individual patients. So if I think about heterogeneity, I think about heterogeneity in, for instance, the pathophysiology of depression. We know, if we talk about the pathophysiology of depression, that there are various known processes involved. Some of those processes are predominantly central processes, but other processes are more involving peripheral in connection with central processes. Think about immune system dysfunction, gut microbiome interactions, but also dysregulations of the HPA axis, for instance. And I think a very clear indication of the fact that the nature of the pathophysiology of depression is so diverse is actually coming from genetics. I think here on this slide you see an overview of the last 15 years of genetic research, genome-wide research. And some of you may have seen the recent Cell paper that came out actually 2 days ago with the latest findings from huge efforts to combine data from depressed and healthy control patients that now really arises at numerous hits, more than almost 700 genome-wide significant hits. And a polygenic risk prediction that is becoming better and better and is now close to 6% explained variance. But more importantly, these genetic findings really illustrate the polygenic basis of depression, indicating a polybiology basis of depression. So on one hand, if you analyze the genome-wide significant findings of GWAS studies on depression, as shown, for instance, on the left in the Howard paper published in Nature Neuroscience a few years ago, these findings clearly point towards involvement of brain processes and central nervous system genetic pathways that are clearly enriched in a GWAS of depression. So really, again, pointing towards central processes. However, that's not the entire story. On the right side, you see a recent paper by Bergstedt et al. And in that paper, they actually looked at the genetic overlap at the level of the genetic variance, and showing here Venn diagrams based on mixer results that shows the number of causal genetic variants that are linked to depression and show overlap with cardiometabolic processes such as CRP, systolic blood pressure, HDL cholesterol, BMI, so all kinds of cardiovascular risk factors. And as you can see from those Venn diagrams, for lots of these phenotypes, there's actually quite a large overlap between the genetic variants that are determining depression risk, but also these cardiovascular risk factor risks. This is also what we study in the Netherlands study of depression and anxiety that I'm leading now for the last 20 years. This is a huge cohort study with multiple waves where we have purposely over-recruited people with depression and anxiety disorders, but also have included healthy controls and are now extending to family members and are in the process of recruiting their offspring right now. In this study, it's also very clear that if you look at people with depression and you compare them with people without depression, there are various clear links to illustrating poorer somatic health in people with depression. That's visible if you look at metabolic syndrome, risk of diabetes, but also subclinical or clinical cardiovascular disease, and even advanced biological aging as determined by higher epigenetic age or higher telomere length. In line with this, if we zoom in within our NESTA study, examining in almost 3,000 individuals, the link between depression status and immune system function, then we also can clearly confirm at various levels of biological layers, looking at biomarkers in the blood, looking at inflammatory genetic pathways at the gene expression level, and also looking at the DNA methylation level, that there is ample evidence that people with depression have higher biomarker levels of inflammation, for instance, by simple C-reactive protein levels that are increased, but also more specific inflammatory marker levels such as IL-6, TNF-alpha, or IL-8. We see evidence of enriched gene expression of immune pathways, and we see enriched genetic pathways of immune signaling. If we look at metabolic alterations, NESTA was involved in a meta-analysis involving 7 additional studies where we gathered data looking at simple blood measures and analyzing 51 metabolites, all very systematically and analyzed it in a similar way across cohorts. We were able to compare more than 5,000 depressed cases and many more healthy controls. And as you can see from this slide, of the 51 metabolites that we measured, 21 metabolites showed significant associations with depression, confirming that people with depression had lower high-density lipoprotein levels, so the healthy lipoprotein, had higher levels of low-density lipoprotein, had higher triglyceride particles, and had higher levels of glycoprotein acetyls, which is an inflammatory indicator. So again, there is a quite consistent and strong evidence that the metabolic profile of depression is different as compared to healthy controls. We just recently even applied larger scale metabolomics, where we are not measuring 52 metabolites, but now are looking at 120 untargeted metabolites, looking at a much broader metabolite profile. And by doing that in 2 waves, we were also able to really consistently look at associations across different time points. And we were able to confirm that in the end, 34 metabolites were strongly, significantly linked to depression status. These indicated downregulation of metabolites that were enriched with long-chain monounsaturated and saturated fatty acids, and upregulations of metabolites enriched for lysophospholipids. And lysophospholipids are activate specific G protein coupled receptors. They are regulators of cellular stress signaling and cell growth and survival. And then you can still wonder, these are all associations with blood markers. Is this truly causality, or can this just be the consequence of depression? And for that, we also can use genetic tools. So we can actually identify through Mendelian randomization whether there is any evidence from a genetic level that genes relevant for these metabolites are then also linked to depression. That's what we did. And by applying Mendelian randomization, we were able to confirm that especially for the lysophospholipid 1-linoleol GPA, we did confirm a higher risk of depression when people had genetically predicted high levels of this gene. And this gene actually is very relevant for determining the fatty acid desaturation enzyme FATs levels, which is a very important enzyme responsible for more than 100 PUFA-containing phospholipoproteins. And these are molecular species really relevant for innate immunity, energy homeostasis, and brain development. Also, when looking at our metabolomic data, we actually were able to look in several papers on the role of acylcarnitins. And acylcarnitins are biogenic compounds that are really central in the mitochondrial machinery. And dysregulations of acylcarnitins have been linked to impaired beta oxidation of fatty acids, and thereby really reducing the mitochondrial energy production. And I think this is really a very interesting mechanism, because we know that the energy levels in lots of our psychiatric patients are generally really rather low. So again, here in Mendelian randomization analysis, we were not only able to confirm levels between the biomarkers and depression, but also confirmed this using genetic data. So I think this all links together quite consistently, and there are now multiple studies, meta-analysis, indicating that these biomarkers and these pathophysiological mechanisms are linked to depression. So it's not just our studies, there are several studies in the literature and meta-analysis confirming such links. However, I think it's important to point out that the pooled effect sizes of all these pathophysiological findings for depression overall are very low. The effect sizes are generally not larger than 0.3. In addition, there is huge heterogeneity across studies. And I think this heterogeneity obviously can be due to differences in methods or designs that studies have used, but I think it can also really be due to the fact that specific depression populations are not always very similar. And that brings me to the point of heterogeneity of symptoms. I think depression is very much known because of the heterogeneity of its symptoms. If you just consider the core symptoms of depression, there are lots of combinations possible. And in depression, some of these symptoms actually even have two directions. We do see patients that have hypersomnia, but also patients that have insomnia, or patients that have hyperphagia or have a loss of appetite. So it's almost impossible, I think, that the pathophysiological mechanism underlying these opposite ends of a symptom spectrum are the same. And these are just only the core symptoms according to the DSM. If you would ask patients, they would identify even much more symptoms that they consider to be relevant for expressing what depression is to them. This is a paper done by patients a few years ago in Lancet Psychiatry, indicating what for them would be the major features of depression. And they really wondered and raised the question whether the variability of all the symptom spectrums that they observe in depression, that leads to the question of whether depression is truly a single disorder with shades of severity and multiple presentations, or whether it is a common name for a number of loosely related problems. And I think these are relevant aspects that we need to consider. And again, I think in our research, we can really truly test some of this. That's at least what we have focused on, also using data from the NESTA project, because what we actually have is quite a lot of detailed assessment at all the waves of the specific symptoms that people reported. And when we dived into the question, well, what are symptom profiles that are relevant? We actually started with latent class analysis. With latent class analysis is a data-driven approach where we look at symptoms and do cluster people that look more alike in the reporting of their symptoms. And when doing that, we were distinguishing three groups. One group, the green line, is a little bit a less severe group of patients, but the other two groups of patients were as severely depressed. They had as many symptoms and scored similarly on the mean depression severity and functioning that we observed in these people. But they basically had different types of symptoms. One we considered the more typical depression group, and the other one the more atypical depression group. Typical depression group was about half of the population, and atypical was about a quarter of the population. And if you look at what are then the typical and the atypical symptoms that stand out, it are truly very often the neurovegetative symptoms that are discriminating here. So atypical depression people were people that had more appetite and weight, had lead-in paralysis, had fatigue, and had hypersomnia. Whereas typical depressed people had less appetite, weight, insomnia, and most suicidal ideation. We also did observe some differences in the correlates. Smoking, negative life events, and childhood trauma was a little bit more prevalent in typical depression, and female gender and early onset was more prevalent in atypical depression. And it were actually the latter group, the atypical depression group, where we saw most obesity and most metabolic syndrome. So that raised really the question, are these two groups also having distinct pathophysiology? So we basically went back to several of the biomarkers I already showed you about that we have measured in NESTA. And we were, for instance, able to illustrate in the molecular psychiatry paper already 13 years ago that people with atypical depression were the ones who had most evidence of inflammation. We did not see this as consistently for people with typical symptoms of depression. Also, when we went and looked at inflammatory gene expression, we could again confirm that the significant enrichment of gene expression in inflammatory pathways was mainly driven by people with atypical depression. 32 of the genetic pathways were significantly linked to atypical depression, and only 2 with typical. Also, when we look at more detailed proteomic signature, we also were able to discriminate between typical and atypical depression by just looking at some of the proteins. Again, identifying CRP, insulin, and leptin being much more increased in people with atypical depression. And finally, also the lipidomic picture I showed you before. If we reanalyze this now, distinguishing between people with high levels of atypical depression symptoms, so hypersomnia, fatigue, lead-in paralysis, having high appetite or high weight, it were exactly those symptoms that showed much stronger and more significant associations with the lipidomic signature. And only 2 significant associations out of the 51 were found when we looked at all other type symptoms and made a sum score out of those. So again, illustrating that it are the atypical energy-related symptoms that are driving the link to the unfavorable lipid profile that we see in depressed people. We also confirmed using genetics that if you, for instance, calculate a polygenomic score based on the genome-wide risk alleles for a specific phenotype, and you do that, for instance, for body mass index, for CRP, and for leptin, that the genetic vulnerability for those cardiometabolic phenotypes is more strongly related to atypical depression and specific atypical symptoms, and not so strongly to typical depression. So to put it all together, this picture is really combining different levels of data now, from the genetics to the pathophysiology to the symptoms. And that arises at this picture of immunometabolic depression that we have recently provided an overview of in Lancet Regional Health Europe, just came out last week, that really gives a very broad overarching picture of the findings I just described to you, where we summarize that people with immunometabolic depression has evidence of immune dysregulation, systemic inflammation, have an abnormal metabolic profile, and that mainly in combination with having high atypical energy-related symptoms. We've also published that people with immunometabolic depression have slightly lower response to standard antidepressants by looking at some antidepressant RCTs. And we are currently testing, as have some other people shown, testing the hypothesis that people with immunometabolic depression may be more responsive to anti-inflammatories. So we're currently conducting a study, a celecoxib study, where we are only recruiting people with evidence of immunometabolic depression, so not all depressed people, and testing specifically in that subgroup whether celecoxib provides beneficial effects. And finally, we're also investing in studies where we look at lifestyle, because I think also we know lifestyle is very important for immunometabolic dysregulations. We're testing whether, for instance, running therapy is of added value for maybe especially this immunometabolic depression group. With that, I want to come to my take-home messages. First of all, I think it's important to embrace depression's heterogeneity in clinical care and also in research. I think I've just only shown you one example coming from our line of research, illustrating that immunometabolic depression may be a relevant subdimension to zoom in on, also hopefully leading to future personalized precision treatment strategies for this subgroup of patients. And I think it's really important to emphasize that this is clearly not the only angle to precision psychiatry. And I want to raise awareness for the fact that at ECMP, we are organizing in actually a few days a precision psychiatry meeting where we really advocate for coming together as a field and really coming up actually with a precision psychiatry roadmap for the future to really take this field further and come up with better clues and better evidence bases to take on for precision psychiatry. And with that, and thanking my team, I really thank you for your attention.
Thanks so much, Brenda. That was great. We have time for a question or two. I see Dr. Fiedorowicz has a question. Go ahead, Jess.
Yeah. Thanks so much for this talk. We know that atypical depression is a more common feature in depression and bipolar disorder, and I was wondering if you had thoughts about how this sort of immunometabolic profile may fit in with bipolar disorder.
Yeah, I think this is really a relevant question. I really hope, and I really am very wishful to be collaborating with people that have, databases in bipolar patients where we can actually look at exactly the same. So I think this is something that we should really extend cross disorder. I don't think immunometabolic depression has to be restricted to major depressive disorder. In our cohort, unfortunately, we only have a few bipolar people, and those are the people that switched over time into bipolar disorder. We have some evidence that those people also have more immunometabolic symptoms. So I think that's clearly a lead, and you're right that that could be a strong link. But I think we really should apply some of the analysis to hopefully some people here that have data on specific depression profile and immunometabolic dysregulation. And then I think we can extend it to other cohorts and patient populations.
Maybe I'll ask one, Brenda, just to extend on that overall theme. I know that it was an adult cohort largely, but We have folks here with expertise in geriatric bipolar, early onset bipolar, mood disorders. What are your thoughts about developmental life stages as they relate to these interrelationships?
Yeah, I think this is really key. I know I have been involved in collaboration with Australia, with Michael Burke and Lianne Smile, for instance. They have young cohorts, and we have published that also in Molecular Psychiatry. They have really confirmed very similar associations and various similar types of symptoms that are also relevant already at very young age with immunometabolic dysregulation. And I think lots of people think that immunometabolic dysregulations arise at like late age. But no, these associations can be found already at very young age, actually. And I think it's really logical because it already starts with a genetic vulnerability, so it should not be only there when people develop somatic conditions. I think these are traits and vulnerabilities that are relevant already very early in life.
I agree with you. So thanks very much. I think we'll take that juncture. Folks have other questions, please feel free to drop them in the chat, and then maybe we can send them to you, Brenda, or you can answer them as we go. But we'll turn to Dr. Jess Fiedorowicz, who's a professor in the Department of Psychiatry and School of Epidemiology and Public Health at University of Ottawa. He serves as head and chief of the Department of Mental Health at the Ottawa Hospital, and his research has been centered around cardiovascular disease and suicide and bipolar disorder, which incidentally are the two leading causes of mortality in bipolar disorder and excess mortality. Circadian function has relevance to these areas, and that's the theme of his presentation today. Not yet published findings from his clinical trial of a chronotherapy for mania. And I'll just add a couple of comments because Jesse sent me a thin bio, probably on purpose, but Jess is also, I think, a highly regarded medical leader, psychiatrist, clinical psychiatrist, and mentor, as well as being a researcher. And we serve together as co-chairs of the International Society for Bipolar Disorders Vascular Task Force. So Brenda, we're going to take you up on your statement of interest. So over to you, Jess.
Yes, we certainly will. Let me share my screen here real quick. And then if I could just get verification that it's coming through okay. Are you guys seeing this? So the presenter view, or are you seeing just the slides?
We see the correct view and hear you clearly, Jess.
Great, perfect, sounds good. So I'm going to present results from the Outer Sunglasses at Night for Mania study. This was presented at ISBD, but otherwise hasn't been presented. So if you didn't see me at ISBD, this is your first chance to see these unpublished results. I have no disclosures. This was funded through the Bazooki Brain Research Fund and the Milken Institute. Was funded as a one-year study. We actually ran it with some startup funds and some personal funds for 2 years. My research is all funded from private foundations or government. I don't do any industry work. Dark therapy case. So this was a case that I wasn't familiar with until an ISBD presentation by Tone Hendrickson. This was an engineer, middle-aged, with bipolar 1 disorder who was quite treatment refractory, didn't respond to many treatments, and had really profound circadian disruption with his illness, including phase advances in mania, which are classic for mania, and phase delays in depression. They tried him on a trial of enforced darkness, and by darkness meaning like he was in a zero light condition for 14 hours from 6 p.m. to 8 a.m. And you can imagine how difficult that would be to do. But strikingly, this actually stabilized both his mood disorder and the circadian disruption, and they were able to sort of titrate down the enforced darkness from 14 to 10 hours. This is from the publication of the dark therapy case. In this lower right here, you see the period of the extended darkness, and you can see that the sleep patterns both become more consistent and stable with a certain period of day, and you can see improved stability of mood, and then activity patterns during that, which was prior to this was quite chaotic. A year after I graduated from medical school, melanopsin was discovered in these intrinsically photosensitive retinal ganglion cells. Melanopsin was discovered earlier, but these intrinsically photosensitive retinal ganglion cells were discovered that seem to be playing a key role in these circadian circuits and helping our brain identify when it's daytime or nighttime. And melanopsin can be hijacked by artificial lighting, such that and there's some evidence that those with bipolar disorder may be more sensitive, but that even low levels of natural light may prevent the brain from essentially recognizing that it's nighttime by signaling melanopsin. And so this led Tone Henriksen in Norway to come up with the idea that could we, now that we know sort of the key mechanism for setting circadian rhythms with the melanopsin signaling in these intrinsically photosensitive retinal ganglion cells, could we block blue light with blue blocking glasses instead of forcing someone to go into a treatment, which I think would be not feasible for anyone except for this, fairly motivated and treatment refractory engineer. With blue blocking glasses, the idea is you could still read, you could still go about your daily life, but we could be letting the brain know that it's nighttime. And so she did a 1-week trial of blue blocking glasses. Raters were not blinded. Participants were not told about the hypotheses and were told that both the clear lenses and the blue blocking glasses might be treatment. So there was an attempt to blind participants. And this showed dramatically large improvements in mania by the Young Mania scale, as you can see here in this figure. The blue blocking glasses were pretty well tolerated. There were 2 in the blue blocking group that had development of depression. They adjusted the timing of the blue blocking glasses so it was reversed. And then 1 person with headache, which was something that they also saw in healthy controls as well as fatigue. So I was had been very interested and wrote a large number of grants trying to get this funded, and thankful still to the Bazuki Brain Research Fund and the Milken to allow us to do this study after many attempts to do this. We conducted a 2-week blinded randomized control trial. We did this at 2 sites here at the Ottawa Hospital. We have 2 campuses that are similarly sized, similarly resources under the same administration and policies. And we followed participants in the trial until up to 2 weeks or until discharge from the study. We were looking at comparing effectiveness with the Young Mania rating scales. The mechanistic data is only in a very small subset and won't be presented here. And then we did some qualitative interviews to try to help see how we might be able to inform implementation of this treatment. We included those 18 to 70 with DSM-5 defined manic symptoms that could provide consent. The study was done in English or French. It had to be willing to provide informed consent. We excluded those with TBI, sleep apnea, or any eye disease or trauma, with the idea that anything that was disrupting sort of the relevant circuits here that we would not want to include. The intervention is on the left. Our control, instead of clear lenses, as was done in the Henriksen study, we did lightly tinted lenses. And you'll see here that both of those block about 10% of visible light. But then for the blue blocking glasses, when you get in this blue spectrum, and I have highlighted over there where the melanopsin wavelength range is, we're completely blocking. Where with the lightly tinted, we're blocking ultraviolet, but we're blocking only 10% of blue. But there is some blocking of blue light with the lightly tinted control lenses. Our primary outcome was Young Mania rating scale. We had 3 assessors that had inter-rater reliability training achieved with ICCs in around the 0.9 range. We attempted to blind participants by or mask participants by telling them we were studying 2 different types of light filters without more detail presented, and as if both lenses were of interest. And then we assessed the integrity of the blind by asking both our raters and participants. The raters were not informed of the study group by the end of the study, and we did several things to inform raters, because raters use information, for instance, from nursing records, etc. And so our research team would review clinical records in advance to make sure that there was no mention of light. Nurses and staff were instructed to record whether participants were wearing glasses or not, but not to mention the color of the glasses in any notes. And they did a generally good job of that. There were just a couple notes that had to be redacted and that were caught in advance of seeing. And then our data analyst was also blinded for the primary and most of the secondary analyses until they became unblinded. So the data analyst just said Group A and Group B. Primary outcome was change in Young Mania. We also looked at antipsychotic use or benzodiazepines with the idea that people were getting treatment as usual apart from the glasses, and that perhaps if the glasses were affected, people would need lower doses of antipsychotics or fewer PRNs of antipsychotics and lorazepams or benzos, which we measured in lorazepam equivalents. I looked at total number of medications and sleep efficiency from actigraphy. Initially, that was only in the sub-study where we did dim light melatonin onset, but our engagement and participation rate in the dim light melatonin onset study was relatively low. So we halfway through the study, we switched the actigraphy to be in the whole sample, and we should have done that from the beginning. And then we had a variety of other secondary outcomes that were registered, but that weren't on the original grant secondary outcome list listed here. The key thing I want to highlight in this slide is that for the Young Mania, the Hamilton, and the Columbia, we did these quite frequently, every couple days, so 3 times a week. But all the other measures were just done at baseline week 1 and week 2. Ultimately, we randomized 42 participants, 20 to the blue blocking glasses and 22 to the lightly tinted control glasses. And you can see here that that randomization wasn't perfect in this smaller sample, and so you can see here that our lightly tinted group is a bit older, more likely to be female, and then I think the other key is that they were less likely to be married, and so the groups weren't sort of perfectly balanced on those variables, but it was a random sample. Age of mood onset, number of hospitalizations, and baseline Young Mania, fortunately, were all nicely balanced between groups with randomization. So we asked at each study visit, the research team would ask the participants about how often they forgot to wear their glasses, and we tried to do this in a very non-judgmental way to allow them to report. We also asked nurses to record whether or not participants were wearing glasses. And then our research team, so the assistant research coordinator, would make an assessment at the end of study about the level of adherence, and we had sort of metrics for each of these. So like fully adherent would be only missing like less than like 10 minutes a day or so, etc. And what you'll see here is that adherence was actually, with reminders, was pretty good, because nurses were also instructed to give reminders, and we gave reminders and talked with participants about like setting an alarm on their phone to help them remember. But adherence was pretty good. I would say 82%, which were mostly adherent or better in the study. We also asked participants whether they thought they were getting an active treatment or not. And so, did you think you were getting an active intervention, yes or no? And what you'll see here is that that there's no statistically significant differences, but we do see more green in the blue blocking group, with more people agreeing or strongly agreeing to thinking that they got an active intervention. Now, we don't know if that's because they sort of found out the hypothesis, or that just the orange-amber lenses look more weird, so you're more likely to assume it's effective, or that they were noticing some sort of effects of it. But there is some violation of the integrity of the blind here. For raters, we did not see that, so raters did not know what participants were getting in general. Sometimes they had some opinions, but they weren't more likely than you would expect in each group, with some disagreement or agreement being similarly balanced. We did have a few situations where the raters became unblinded, and we then switched raters, so that way we had an unblinded rater throughout. And here's our primary result. So we did not see any differences between the two groups with the blue blocking glasses compared to the lightly controlled glasses. And this was not actually even remotely statistically significant. Our p-value was 0.9 here. Hamilton Depression Rating Scale was something we were checking for safety, given concerns for depression. I want to point out that at visits 5 and 6, the sample is quite small. So we had a lot of people seem to be discharged between visit 3 and 4. Visit 3 would be the 1-week mark. So there is this little bit of a signal of worsening depression in the blue blocking group. I think that this isn't clinically meaningful, and in later slides I'll sort of build that case. Average antipsychotic dose was strikingly consistent between the two groups. So we're looking at 4 100ths of a milligram per day on average in risperidone equivalents, and 4 100ths of a milligram in benzodiazepine equivalents, so strikingly similar. Total med use was quite similar between the two groups. Our sleep efficiency measure we had in only a subsample, and that was also did not look different, although was slightly higher or slightly better in the blue blocking group, but again, not remotely approaching statistical significance in this sample of 16 that we had this data. Other outcomes, we have PMQ-9, DigiBD, and ASRM, all self-reported mania rating scales. No differences in anxiety, depression, sleep quality, and morningness, eveningness questionnaire. We did not see any differences between the two groups here. Adverse events. So I want to highlight, we had an awful lot of headache. And one thing I want to comment on here is that our research team was reviewing the chart and would report a side effect if say, a patient asked for acetaminophen from the nurse for headache. Even if the participant didn't report headache to us in our meetings, we would report that as an adverse event, and we were looking for these sorts of things. And so we had an unusually high headache rate in both groups. But otherwise, really not a lot of differences in adverse events between the two groups, and the total number reported for each group was quite comparable. I want to dive into the depression data a little bit more because there was this signal for greater depression in the blue blocking group. If we look at the final visit Hamilton depression score, we don't see a lot of high scores, and the two scores that we do see that are higher are actually in the lightly tinted group. So I don't think that the finding that we were seeing there in the final end of the study was being driven by people with really high depression scores. And you may recall that the scores for both groups were relatively low, and I was actually expecting to see more depression by end of study visit. Just keeping an eye on the time here, we're still good. We did qualitative data with semi-structured interviews using a standard script. These were transcribed and analyzed using a grounded theory approach. Two research staff carried that out in parallel, met to consolidate codes, etc. We transcribed interviews from 24 interviews of participants and 10 interviews of staff. And even though this was a negative study, I think the qualitative data was interesting and somewhat encouraging, and I want to share some of it briefly with you before we go to discussion. Some themes that came up from participants were that they were motivated to participate in the study. They thought that there was a patient-centered approach and experience for them. They appreciated being involved and often had some research understanding even before being involved in the study. They had comments, they had opinions on the efficacy of the intervention, many thinking that it worked, some thinking that it didn't work, and they reported some challenges to trying to wear glasses, etc. Overall, like the mind mapping theme suggested that people were ready for this intervention. I would say that the level of interest in this intervention was very high. People were very interested in a non-medication option to treat mania, and were quite interested in glasses in particular as a potential therapy option. So it's unfortunate that our results were negative because there was certainly an appetite for this with participants. Clinicians appreciated the opportunity to be involved with interacting some with the research team, thought that it was positive that patients had this available, thought that this was an acceptable treatment add-on for both them and patients, and did talk about some challenges with having participants, patients that they're taking care of, participating in the study. Overall, though, the input from clinicians was that this was a very acceptable intervention, that they were supportive of this treatment and hopeful. Many also perceived it as being efficacious from their patients that were involved, but as we can see from the RCT data, that did not come through. So again, highlighting the importance of doing controlled trials and not focusing on anecdotal evidence, because the clinicians anecdotally felt that these glasses were helpful. A few qualitative take-homes to highlight before we wrap up for questions. Participants and clinicians liked the treatment. Comment was glasses were cool and it was fun picking out the different styles. So we had the lenses were the same, were similar, different, but we would put them in different frame styles, and so participants could pick styles, including styles that would wrap around or go over existing glasses for those that wore glasses. There was a theme of empowerment, that people felt that they were empowered by the chance to participate, and that they felt like it was even helpful. Even participants that didn't think the glasses were effective felt that participating in the study was helpful for them and had sort of a positive impression. So again, highlighting the idea that the opportunity to participate in studies like this is something that participants value. A couple quotes that I'll share, and then we'll wrap up for questions. Even though they weren't really part of my treatment as a patient, I felt it was another place where I got to talk a bit about what I was going through and was very much listened to. I felt what I had to say was valuable and good for my own processing of why I was here and my diagnosis and all sorts of things. Another person commented on the chance to talk to research staff, talking to people who are very nice and willing to let me go wherever place I'm at, which when I'm manic is very fast. That was very nice because it's like, oh, maybe that's not anything really wrong with me. Maybe I'm not crazy. Maybe I just have a fast processing speed. But I'm still kind of making sense to people. So it seemed like just contact with study staff was helpful to folks. So conclusions, this is the first study with blinded raters of blue blocking glasses for mania. In the setting of treatment as usual, the add-on treatment was not more effective than lightly tinted glasses on our primary outcome. There was a signal for increased depressive symptoms as measured by the late HAM-D in this small subset that were late in the course of the trial. But we didn't see like high scores or categorically reporting depression by participants as a side effect. Participants, clinicians find it acceptable, but unfortunately it doesn't appear to have worked, at least in this study. So some acknowledgments. Joe and Jessica were key in running the trial, so they were unblinded throughout and ensured that we stayed blinded, and here they are sporting their sunglasses. They also led and conducted the qualitative analyses, and Jessica is the first author on that paper coming out. Our raters were all physicians, Eric, Marco, and myself, and so I want to give special props to them because we often had to, when we recruited a participant with mania, it was something that we needed to run with and get going with right away, and so appreciate them, and we often had to make last-minute changes in our schedules to accommodate the study assessments. And then finally to my PhD student at Epi, Sarah Siddiqui, who was carried out the analyses, and which was blinded for primary and got unblinded at some point during the secondary analyses, but didn't matter because they were all negative essentially. And this is the whole team. So I see we have a question already from Benoit, and I don't know if there are other questions, but why don't we go ahead and take that question right away?
So thank you for a great presentation, very fast. Disappointing result, but going back to your conclusion slide, maybe it was, and I'm not mean to be critical, it didn't work, we all have more failed trials than successful, but maybe it was the wrong design. What you have shown is when people get 7 milligrams of risperidone per day, the blue blocking glass had nothing. Okay, maybe the medication worked enough over 2 weeks. So I'm wondering, should it be done for people who don't want to medication, or should it be done?
Oh, that's an interesting point.
Should it be done for, there is a question from Andrea Vasiliev who say, should people, given there is no risk, should it still be offered? Maybe if a patient doesn't want to take medication. And then my real question, because I'm preoccupied, we all do and are obsessed about acute treatment in psychiatry, and we don't do enough prevention. Could it be used for people who are at high risk for a switch to mania? Patient with bipolar, who is not manic yet, but who have poor sleep. They come and they tell you, the past few nights, or my thoughts are racing. So they are symptomatic or pre-symptomatic. So anyway, you have more.
No, that's a fantastic. Those are all fantastic questions. I really appreciate them. Let me sort of take them one by one as I remember them. So it's always very hard to show an active treatment on top of another active treatment, right? And certainly these groups were relatively aggressively treated for their mania in the inpatient setting, and so there's, could there be risk of type 1 error when you have the background of relatively high doses of antipsychotics and other medications being prescribed? Type 2 error is also a risk in that we recruited 42 participants. Our target was 51. So certainly type 2 error is also possible in that way. Certainly it suggests that that we're not seeing much of a signal in those that are already receiving a fairly active treatment, but identifying a subgroup such as you mentioned, like those that don't want to take meds, or for prevention, I think would be really an especially interesting study. I hope that this doesn't nix people's opportunities to get funding to do further work in this area, because I think this is a very biologically plausible mechanism. We know that other circadian therapies like light therapies are very effective, and so I would love to see if this could be, still studied in some way. And I think prevention might be, I imagine it's both of those are hard studies to do, right? To get a sample of patients not taking meds is going to be harder. Prevention studies always require, decent sample sizes and being able to select high-risk groups. But if we can identify high-risk groups, it's not a costly intervention. The glasses are not expensive. Once we give people the glasses, they could use them for a fair amount of time. So I think that prevention study would be fascinating. So thank you for those questions. Yeah, and if anyone is interested in collaborating in something like that too, please don't hesitate to reach out. I would love to find some way that we can still maybe look at this, although it was certainly discouraging to get a result that was so clearly negative on the primary, for sure.
That's just a comment and a question. The comment is, I think there's differences of opinion about preliminary or pilot studies, but I think to my view, you achieved the purpose of it, which is you got people to wear glasses and you kept a good blind integrity. And I don't think we can expect 30 people to tell us what we would find with 300 people. So anyway, we can chat more offline, but
yeah.
There's so much variability, and I think
there's definitely interest. Like, people really were interested in non-medication options, and we had several people that were like really upset with their meds, but were totally engaged with the glasses.
Yeah, I mean, I think Benoit mentioned the 7 milligrams of risperidone, but the comments from the participants themselves were the non-specific support of the staff. So it is really you're having to jump over 2 fences, not 1 fence. My question was about the blinding. Is it possible to get these glasses made so that they're identical?
Yeah, it's not. So if they're blue blocking, they're going to have the color tint, and so yeah. So that was a common questions we're going through, but unfortunately, that's a barrier that we have now. There could be possibilities, like especially if we're focusing on more of an efficacy than an effectiveness study. There could be ways to manipulate environmental light within a unit. If we wanted to sort of do some sort of proof of concept or something like that, in which case I think it might not be super obvious to participants if the light's being manipulated in some way. But the true blue blocking glasses are going to be orange or amber colored. Fortunately for us, for helping with the blinding too, though, there are a lot of glasses, like I'm pretty sure these, that are marketed as blue blocking, but they just have like a slight blue, supposedly to help with glare from computer screens. But my understanding I'm not an expert there, but my understanding is that's actually not effective, but a common marketing sort of gimmick.
Thanks, Jess. I see Brenda has a question.
Yeah.
Very interesting. I was wondering, in hindsight, do you have any idea why your first data that you showed were so positive? Was there something specific about that population? I mean, any clue, or is it just chance?
So a few differences. So one is that the initial Henriksen study, the raters knew which group people were in, and so they weren't blinded raters, and we know that can introduce bias that can be hard to quantify. They also use clear lenses, and so if there is some biological effect of just blocking 10%, that would also reduce our ability to see effect. Our sample had higher mania scores at baseline as well, and so I want to say, I'm trying to remember off the top of my head, but I want to say we're looking at maybe like a 3 to 5 point difference in the young mania at baseline. So our patients were a little more manic, and so perhaps that played a role. It was also done at a much higher latitude. I don't know if that matters or not, but yeah.
Okay, well, that brings us to our last minute. So thank you both for being on time, comprehensive, engaging, and open with the Q&A. And just invite folks to join us again March 13th, looking at consortium approaches to bipolar disorder with Kate Burdick and Ole Andreasen. And thank you all for attending today. Have a great rest of the week.
Great, thanks.